Hi, this is Emma Dowd, again, the Assistant Director of the IUSM Proteomics Core, and this segment is going to talk about scaffold analysis and visualization. So Scaffold is a software from Proteome Software, and here is a link here, and it is a tool specifically designed to help visualize and share data. So this is a picture from the page, and you can go here to download Scaffold as a viewer. So you do need to purchase a license if you are planning on making Scaffold files, but to view the data or a Scaffold file that we have produced or someone else, you can just download the software from this site. So again, just to loop this back around, it's one of several ways of visualizing results from the different tandem mass spectra protein peptide search engines. So Scaffold really will let you take any search engine results and load them. It will allow you to apply FDR cutoffs from within the while it's loading the data, or it can now you do an X tandem search that's actually integrated in the Scaffold software. But again, this is only if you're creating your own Scaffold file. And it will allow you to help with your data analysis. So I'm going to walk you through how we create Scaffold files to send and share to viewers. So I recommend you start off with some of the other modules in this segment, and specifically Proteome Discover, because that's generally where we're doing our deeper data analysis, running the search engine and doing FDR cutoffs. However, what we can do is we can take the results from Proteome Discover, and in addition to just exporting an Excel file directly from Proteome Discover, we can upload the data to Scaffold, and it's a pretty easy way for you to have a viewer that will allow you to examine all of the data that we've used for your experiment. So you can really dig in a little deeper without having the more in-depth knowledge that Proteome Discover might require. So when you're creating a Scaffold file, you just go to Scaffold, new project, and as you add each sample, essentially what you're doing is you're queuing the peaks or results file from your search engine. So that can be from any specific search engine, but in this case, it is a file that Proteome Discover makes, and it's generally in the same folder as the project folder for your Proteome Discover. You will need to, when you load and analyze the data, have the same FASTA database that you were using as you did the search. I'm just going to get the highlighter. So in this case, a human UniProt database with contaminants present. And so you can, again, I recommend you listen to the lecture by Dr. Mosley covering FASTA databases if you haven't. You now have an option to analyze with X-Tandem. That's a different search engine. And there's a few different scoring systems, and this is essentially the different FDR measurements. So LFDR is this all instruments cutoff. Peptide profit is originally an academic false discovery rate algorithm. And you can also now use this pre-filtered mode, which accepts the results from Percolator or whichever search engine, if it included a cutoff or a specific score around this. And so this is nice because if I was able to use Percolator in my Proteome Discover, I can directly use this. Now, what does happen is this is an additional cutoff on top of whatever cutoff that Proteome Discover or your search engine had. So you do have to be a little bit careful when you're looking at this data, but it usually works out pretty well. So you can do this standard grouping, pre-compute FDR thresholds, and you load all your data. And what's really nice is you get this great viewer where you can really filter the data yourself. So it's definitely just a little easier to see everything than it is in Excel. It can get challenging if you're doing a larger experiment. We don't really recommend, it's a little challenging to use Scaffold for that. But if you're doing several, in-gel digestions, or you're comparing a bunch of different IP or AP experiments, Scaffold is a great tool. And so I'm just going to walk through some of the functionalities of the viewer. So after it's saved, you can share, view, filter, and it doesn't matter what I did, you can do it to the file after creation. And so really, it's really important to look at all of these display options and kind of understand what it is you're looking at. So Right now, the display option is set to total unique spectrum count, but if you drop this down, you can filter for exclusive spectrum count, total spectrum count, not unique, any number of different, essentially abundance equivalents. You can also filter by required modification. So if you only want to look for things that have a phosphorylation on them, you can drop down this filter and immediately pull out any proteins that have spectra that had confident phosphorylations. You'll also see that you have these are the filters up here, the protein, minimum peptides, and peptide threshold. So right now we only have a minimum of 1 unique peptide. Sometimes people will knock that up to 2, but it's perfectly acceptable to leave it at 1. And essentially the peptide and protein thresholds are your false discovery rates. And I recommend you load your data and kind of play around and see how it looks. These are our defaults for sharing with folks, but there's lots of different options. So see in this view, then you will see all of these proteins which have been identified, their accession numbers, molecular weights, whether there's grouping ambiguity, that just means whether there are multiple proteins that could be assigned to that accession number. Again, that gets into splicing variants. And then this lets you view a relative quantitation, essentially. So it's very good for label-free data. So just off the bat, you can see there's a bunch of keratin. And this is really common if you have a gel band specifically, just because you're generally doing a lot of manipulation. Someone has had to cut the gel bound out, extract it several times, and keratin is in the air, it's in dust, it is dust essentially. So you'll see that as a very common contaminant, which we always include in our searches just for improved confidence of identifying the correct protein, which is the second hit, this metabotropic glutamate receptor 5. So you can see it's equal amounts, but if for an in-gel digest, that's okay. It gives us good data. So on the left here, you have your load data. That was your setting up your experiment. This is the samples view. You have a proteins view that in which if you click on any one of these proteins and go to protein view, if you click on proteins, you can also do filters for specific proteins. So that's kind of handy. This is definitely a lot more simplistic than any of the filtering you could do in Proteome Discoverer. And then when you go to this protein view, you will see all the coverage you had of that specific protein. And this is great. And this, I think that it's underutilized when we send data to people. There are definitely people who walk through this data step by step. But here you can see each peptide that was identified in each sample. You can search and filter for any specific modifications on any peptides, and you can actually view the individual spectrum and fragmentation that was identified for each peptide. So here you can see the FASTA sequence and the overall coverage in this specific sample. So if you click down, you have a wild type versus a knockout. And you can click through these, and each one would show you different yellow highlights, which indicate peptides that were confidently identified by the FDR cutoffs that were specified. If you click on any of these individual peptides, it will link to the sequence and MSMS information listed here in this chart. And this is great because you can actually see your Sequest X-score and delta CN. So this was discussed in Dr. Mosley's Sequest lecture. And because it's really nice to be able to sort and view yourself the quality of each spectra for each peptide. Any of the highlighted residues that are in green just mean there was a modification found. So in most of these cases, it's methionine oxidation, which is a +16. And that's really common for any highly processed sample. Methionines just oxidize really easily. So what's even better is if you click on some of these peptides, you can walk through at this tabular level and view the specific spectrum, spectrum model error, and fragmentation. And that's really useful if you're trying to get some figures for papers as well. So for example, here's a slight zoom in where I've selected this wild type sample. I selected this very long peptide with a really high Sequest score. You might remember from Professor Mosley's lecture that the longer peptides do have a tendency to have quite high scores in Sequest. And then down here, instead of in the protein sequence, which had the highlights, if you select the spectrum, you can actually see all of the B and Y ions that were assigned to this sequence. And this is just really useful. If you click spectrum model error, that will give you the mass error of each of the fragments. Here you can see exactly what the precursor ion was. And so it was a +3 charged 969.77, and it was about 2.3 ppm off from the parent error. So a little higher than we like, but because of these excellent fragmentation patterns, we can confidently identify this peptide. So if you would like statistical significance to your samples, so this is perhaps to quantify or qualify an IP or an AP, if you want to say that you have confidently identified potential interactors of a protein that you're pulling down, there are some different spectral quantification methods that are available. So this is different from the quantification that we'll talk about next week in mass spec quantitation. I think we'll mostly be focusing on precursor quant as well as TMT or SILAC quantitation, which are really precise methods. So spectral counting is kind of a little bit of an older method, but it does give you significant results in terms of whether something is present or absent. So it's very fast, it's a little bit more simplistic, and it will give you the statistical evidence you need if you're trying to just make a claim that something is an interactor. For example, when you're validating an antibody or an IP method. And when you go to this quantitative analysis setup, you can see that if you just have 2 replicates, you only have the choice of a Fisher's exact test. If you have more replicates, you could do something like an ANOVA or a t-test. So in this case, we do often run a Benjamin-Hochberg correction of a Fisher's exact test, looking for significance of less than 0.05. And so this is an example comparing 2 samples pulling down a protein of interest. And then you're looking for significance. And so again, this is not an optimized system by any means, but you would hope that in your pull down of interest, you have significantly increased your protein of interest. So it would have 28 versus none in a control setting. So this is the sort of results that you can then also be exported into an Excel table and play around with. But yes, this is a simple, quick way to do some quantitation if you don't have too many replicates and don't want to pay for TMT or SILAC labeling experiment. And I hope you can reach out if you have any questions. I'm going to directly go into Scaffold PTM next.